Hey, hello everybody. Can you see me and hear me? Yes? Yes. Good. Beautiful day today. I wish we could have class outside, but not possible. Okay, so what are we going to do today? So last class on Thursday, we discussed how this electrical signal goes through the motor neurons and reaches the muscle fiber. So today, what we're going to do is, how does the muscle fiber reply to this signal that's coming from the motor neuron? And this is what is going to be converted now into muscle contraction. So remember, the muscle fibers, the muscle cells are able to convert chemical energy into mechanical energy. So the movement, how is this possible and how is this achieved? So we get excitation from the muscle fiber, right? This electrical signal that goes down the axon, reaches the axon terminal. Then you have the release of sodium ions. No, you have the release of acetylcholine, the neurotransmitter, that diffuses across the synaptic cleft, binds to its receptor on the muscle fiber, the muscle end plate, and then what happens next? You have sodium channels opening, sodium goes in and changes the potential. So the resting membrane potential is minus 90 millivolts. When sodium goes in and reaches its maximum, now it goes to plus 30 millivolts. So now, having this scenario where you have an electrical stimulus, how is that converted into muscle contraction, into the sliding of the thick and thin filament? So that's the coupling of the excitation by the motor neuron and contraction of the muscle fiber. Okay, so here you have an illustration of this process. So here's the axon and the axon terminal. You have the action potential reaching. And here you have calcium channels. So calcium comes in, floods the synaptic terminal. And this allows or stimulates, triggers the synaptic vesicles to move towards the periphery, diffuse with the plasma membrane, and release acetylcholine. So now the acetylcholine at the motor end plate opens sodium channels, and the sodium channels now cause the sodium goes in to the muscle fiber and causes a change in the potential of the muscle fiber, going from minus 90 to plus 30. So question, they cannot only control oh, this is not a question, sorry. Okay, so here is the muscle fiber. And remember the myofibrils, right? These cylinders that are made up of the thick and thin filaments. That they have the actin and the myosin. Now remember that close to these myofibrils, you have this sarcoplasmic reticulum. And this sarcoplasmic reticulum is highly enriched in calcium. It has 10,000 times more calcium than in the cytoplasm. So you have high concentrations of calcium in the sarcoplasmic reticulum. And this area of the sarcoplasmic reticulum that's close to the T-tubules is known as the triad. Now notice this, that here you have the sarcolemma, which is the plasma membrane of the myofiber. It invaginates inside, so it goes inside the muscle fiber. So this allows for the extracellular fluid to go in. It also allows for the current, the electrical current that is generated by the opening of the sodium channels to be transmitted into the muscle fiber. And this is the beauty of this process. So the change in potential, so that goes down the tubule, the T tubule, sends a signal to the sarcoplasmic reticulum, opens the calcium channels, and now calcium comes out of the sarcoplasmic reticulum and floods the myofibril. And remember that on the myofibril, on the actin filaments, now I'm going to explain this more slowly, but I just want to give an overview now. Yes, plus 30. It goes from minus 90 to plus 30 millivolts. So on the fin filaments, you have that troponin and tropomyosin, and troponin is the complex that binds calcium, and that initiates the process of muscle contraction. So now you see how the change in potential along the sarcolemma is transmitted to the T tubule. And it's this change in potential that sends a message to the sarcoplasmic reticulum to release calcium. Now let's look at this in more detail. So here you have a very beautiful view. This is the sarcolemma, and this is the T tubule. The T tubule, you see, that goes into the muscle fiber. And it's because it's sarcolemma, so it's the plasma membrane also. So the sodium, the action potential that is traveling through the sarcolemma is transmitted through the sarcolemma that's part of the T tubule. So you have an action potential going down here, okay? So there's a change in potential, and it's this change in potential that sends a signal to the sarcoplasmic reticulum. Remember, there's 10,000 times more calcium in this blue structure, the sarcoplasmic reticulum. And you see how it's right above the myofilaments. So you see the thick and thin filament. Okay, so let's go a little bit more complex, this very simple image. So here you have the axon terminal with the synaptic vesicles that contain acetylcholine. Acetylcholine is released into this synaptic cleft, and it diffuses across the synaptic cleft and binds to these channels. And if you remember, these channels that are at the end plate, here's the end plate, the area of the sarcolemma that is in close proximity to the axon terminal. So these channels are different from these. So these here are ligand-gated ion channels. What does that mean? That means that they open when a ligand, in this case it's acetylcholine, binds to them. You have two molecules of acetylcholine that bind to these receptors that are also sodium channels. So when acetylcholine binds to these, they open and sodium goes in. Remember that there's some potassium that also goes out. But here the most important event is entering of the sodium channels, of the sodium ions. Okay, so now, instead of being negative, now the inside is positive, and this signal is transmitted. So this causes a change in potential from minus 90 to approximately minus 60. So these numbers are approximate, okay? So now you go from minus 90, which is the resting membrane potential. The muscles are resting. But now after this acetylcholine opens these channels, it goes from minus 90 to minus 60. And this minus 60 is going to be, this electrical signal is going to be transmitted to the rest of the sarcolemma that is not in close proximity of the axon terminal. So now you have a current of minus 60 millivolts reaching these channels. These sodium channels are not ligand gated. they're voltage gated ion channels. So that means they're closed. And when the resting membrane potential goes from minus 90 to minus 60, they're open up. And now sodium goes in to the muscle fiber. So you have lots of sodium coming in, and the voltage now goes from minus 60 to plus 30 millivolts. Okay, but now you have to envision the current going down into the T-tubule. So this is still the sarcolemma. So this is still the plasma membrane. It's an invagination, an extension of the sarcolemma into the muscle fiber. So you have now the action potential, this electrical current going down the sarcolemma of the T-tubule. So this is the T-tubule. And now you see here, there's a receptor known as dihydropyridine or DHP. And this receptor, when it's activated by the voltage, by the current that's going down the T-tubule, causes these calcium channels that are on the sarcoplasmic reticulum to open. So you see here the events. You have ligand-gated ion channels that allow sodium to go in. Then the current is transmitted to the rest of the sarcolemma, where you have voltage-gated sodium channels that open. And this action potential, this change in potential, this electrical current, travels down the T-tubule, the sarcolemma on the T-tubule. When it reaches this dihydropyridine, that are receptors on the sarcolemma, they send a signal to calcium channels. These calcium channels now open and allow calcium that is inside the sarcoplasmic reticulum to come out. And this calcium now floods the inside, the cytoplasm of the myofibril. Okay, so you have calcium here that induces the movement of the synaptic vesicles towards the plasma membrane so that acetylcholine can be released. And you have calcium here that is being released when there's a current, an action potential coming down. Okay, so these channels that are on the sarcoplasmic reticulum are voltage-gated calcium channels. These are voltage-gated sodium channels. These on the sarcoplasmic reticulum are voltage-gated calcium channels. So all this is incredible. So all this is happening when you're moving, when your muscles are in action. So all this is controlled by the motor neurons that initiate this cascade of events, that now we are at the stage where you have a lot of calcium in the muscle fiber, in the muscle cell. Okay, so here you see in more detail the acetylcholine being released. Remember this, the motor end plate in this area has many folds because you have lots of acetylcholine receptors here. So now you have the action potential being transmitted down the T-tubule. And now here you see the receptor that is DHP, dihydropyridine, and these are all proteins. And what you have to think is that the structure of these proteins changes, the folding of this protein. Here you have a alpha helix. The shape of these proteins changes in response to different stimuli. In this case, it's a voltage that's coming down the T-tubule. Now the signal is transmitted to from the dihydropyridine receptor to the calcium channel. And you see here the sarcoplasmic reticulum filled with calcium. Now the calcium channel, the voltage-gated calcium channel, opens, and calcium, represented by these green little circles, comes out. Can you repeat why there are many folds in the motor end plate? Okay, so here you have many folds because this allows the surface of the sarcolemma is larger, right? If you had straight, or now if you have folds, the same in the intestinal membrane, mucosa. So this allows for many, many more acetylcholine receptors to be present. So that means that the signal is going to be very, very strong because you have all these acetylcholine receptors that are located at the end plate potential, at the end plate to be activated. And this generates a current that is strong enough that is going to be transmitted along the sarcolemma. Okay, is this clear? Okay, so now we reach this point where you have a lot of calcium being released from the sarcoplasmic reticulum. What happens to this calcium now that's in the cytoplasm? Now remember that just below the sarcoplasmic reticulum, you have the myofilaments. So in red here, this is the fin filament, and in bluish or lavender, you have the thick filament with the heads sticking out. Now, the fin filament has troponin, here represented in yellow, it's spherical, and you have the tropomyosin that is covering, when there's no calcium, the tropomyosin, it's a spaghetti-like structure, is covering the actin binding sites on the actin. But now when calcium, here represented by these green circles, binds to troponin, you see troponin binding calcium, it causes a change in position of the tropomyosin. And now the calcium binding sites are open or exposed so that the myosin heads can bind to the calcium. Now remember that the myosin heads are also enzymes. They are ATPases. They can hydrolyze ATP to release energy. So this energy is used to make the cross bridges. We call when the head of the myosin is bound to the actin, we call this a cross bridge. And now, so you have the myosin that is bound to the actin, and now you have to detach the myosin head. So you need another molecule of ATP. And now with that ATP, now the myosin heads are going to come off the actin and they're going to move along the fin filament, along the actin. This is the Z line of the sarcomere. So you will understand this in the next few slides. Okay, so troponin binds the calcium, and tropomyosin, which is covering the actin binding sites, moves. So now the heads of the myosin can bind to actin. So you see here what happens. So the heads of the myosin move along or slide along the actin filament. They bind to one actin, and then with the ATP, they come off and they bind to the next one. Then they come off, they bind to the next one. So they're walking over the actin, and that's the sliding. They're sliding over the actin filament by binding to the actin. And binding, you need one ATP. To come apart, you need another ATP, but they're moving along, they're sliding over the actin filament. So here you have the different steps. So let's start here. You have the myosin and the actin are separated. You have ATP. Now you have the cross bridge form because the head is an ATPase, so use the energy from the ATP to form the cross bridge. And then you have another ATP that allows for the cross bridge to be, for the myosin and the actin to become apart, to separate. So now you have the separation, and now it moves again to the next actin. So you have two ATPs that are necessary for the sliding of the thick and thin filament upon each other. Question: does troponin and tropomyosin do the same thing in terms of helping with muscle action? What's the difference? So troponin binds calcium. When troponin binds calcium, it changes its structure, its folding, that will act on the tropomyosin and triggers the movement of the tropomyosin to allow it to expose the actin binding site. So the two proteins, troponin binds calcium, tropomyosin covers the actin binding sites. So once troponin binds calcium, then causes the tropomyosin to move away from the actin binding sites. Okay, so now you see this in movement. Here's the myosin with the head. It binds ATP, the green, and now it's moving along the actin filament, the thin filament. And here in the purple, this represents the troponin. So when it binds calcium, then you see the tropomyosin, which is represented in blue. It moves away so that the myosin can now bind to actin. And then it comes apart, it binds to the next actin, comes apart, binds to the next segment. And that's how it moves, sliding. The thin and thick filament are able to slide upon each other due to these molecular events. I mean, this is pretty amazing if you think about it. I love to describe this every year because I think it's an amazing process at the molecular level. This is the basis for muscle contraction. Okay, so now once this happens, and you have the sarcomeres coming together, remember the H zone, which contains only thick filaments, is going to be narrower. Now you have to relax the muscle, right? You cannot be always in the contraction state. You have to relax. So what needs to happen? You need to get rid of the calcium ions, so that the troponin is not activated. So that's one major factor. And you need to get rid of acetylcholine, right? Because acetylcholine is the neurotransmitter that initiates these events. So for relaxation to occur, you need two things, two major things. One is get rid of acetylcholine at the synaptic cleft. So there's an enzyme there known as acetylcholinesterase that is going to metabolize acetylcholine. It's going to neutralize. Acetylcholine is no longer able to bind to its receptors. And then you have calcium channels that will bring calcium back into the sarcoplasmic reticulum. And that's how you can induce relaxation. So you see here the sarcoplasmic reticulum. So there's calcium channels. So you have some calcium channels allow calcium to go out. Other calcium channels or calcium pumps allow calcium to go in. So here you need active transport because they're pumps that are pumping calcium from high concentration to low concentration at this state. So you have calcium coming in. Inside the sarcoplasmic reticulum, there's a protein that is very, very avid for calcium. It has a high affinity for calcium. And that protein is known as calsequestrin. So it sequesters calcium. This protein, calsequestrin, is located inside the sarcoplasmic reticulum. So you have calcium coming in to the sarcoplasmic reticulum, binds to calsequestrin. So that's why there's so much calcium inside the sarcoplasmic reticulum, because it can bind to calsequestern. Okay, so that step is taken care of by the calcium pumps and the calsequestern. So now, how do you get rid of acetylcholine? So here you have the enzyme acetylcholinesterase that is located at the synaptic cleft. So you see here now this cartoon shows you this is the motor end plate. So that area of the sarcolemma of the muscle fiber that is in close proximity of the axon terminal. So you see here, this blue represents the acetylcholine receptor, but right here you have acetylcholinesterase. That's very, it's important. It's located in the synaptic cleft. So it breaks apart acetylcholine into choline and acetate. This diffuses out, and choline goes back in to make more acetylcholine. So this enzyme is so important that nowadays the only way to treat, it's not to treat, is to prevent the progression of Alzheimer's are inhibitors of this enzyme. So in Alzheimer's, the neurons that degenerate are neurons that have the neurotransmitter acetylcholine. So that means that in Alzheimer's, there's very, very low levels of acetylcholine at a specific site, the hippocampus. So the hippocampus is the area in the brain that is responsible for short-term memory. So there's short-term memory and long-term memory. Short-term memory is located, the neurons that are responsible for short-term memory are located in the hippocampus. Now, this has nothing to do with muscle contraction. I'm just giving you this knowledge so that you know how important this enzyme that is present not only at the synapse, at the neuromuscular junction, but is also present at the other synapses. So this enzyme is very, very important to break down acetylcholine. Okay, so relaxation. You see now there's another event that is important. So we talked about the opening of the calcium pumps to allow calcium to go back into the sarcoplasmic reticulum. So, but what is the signal now for this to happen? So what happens is, remember that the action potential goes first from minus 60 to plus 30 millivolts. So when it reaches plus 30 millivolts, there are potassium channels that are also located at the sarcolemma. When this action potential reaches plus 30 millivolts, actually a little bit before, the potassium channels open up and potassium goes out. And this allows for the reestablishment of the resting membrane potential. So now from plus 30, it goes down to minus 90 again. That's the resting membrane potential that is required to trigger relaxation, so that the calcium pumps are going to be open when the membrane potential goes back to minus 90 millivolts. So then the calcium pumps open up and calcium goes back into the sarcoplasmic reticulum. So now you see here what happens to the sarcomeres. Remember, there's thousands and thousands of these sarcomeres in one muscle fiber. So when there is contraction, that means that you go from this stage, which is the relaxed stage, you go to the contraction stage, where the H zone that contains only thick filament is very narrow. So compare the width of the H zone here with the width of the H zone here. So the H zone is the area that only contains thick filaments. So here you have the thick and thin filaments overlapping just a little bit, right? So in this muscle contraction, and the thick filament is walking along or sliding along the thin filament, then that means that the H zone narrows, and also the distance between the two Z lines in the sarcomere come closer because the thick filament is moving along. You see here in this slide, it's very, very clear that the size of the thin and thick filaments don't change. What happens is that they slide upon each other, bringing the two sarcomeres closer to each other. So the myofilaments never change in size. Okay, so when the Z lines come closer, the movement, the narrowing of the Z lines develops tension or force. So here you see the Z line, you see the thick filament moving upon the thick filament moving upon the thin filament, bringing the Z lines closer to each other. And this is what causes the development of tension or the development of force. So tension is what happens when the muscle develops force, when the muscle is contracting. It develops tension because there's tension on the Z lines. They're being pushed closer to each other. So that's what develops the force of the muscles that allows the muscle to contract. Okay. So do both the thick and filaments move? Well, it's mostly the thick filament, right? The thick filament is, you see that the thin filament is stationary, attached to the Z line. So it's mostly the thick filament that moves along the thin filament. So now you see how that electrical signal that develops at the motor end plate is transmitted to the membrane of the muscle, to the sarcolemma. So it causes the thick and thin filaments to slide upon each other. And because the sarcomeres now move closer to each other, now this causes the muscle to develop tension, to develop force. Okay, so there's a state in when a person dies that is known as rigor mortis. And that goes on for about 3 or 4 hours after death. And this allows detectives and other people like that to know more or less the time of death of a person. And what is this? What is rigor mortis? Rigor mortis is the state of the body that where the muscles are in full contraction, they're not relaxed. And why does this happen? This happens because when a person dies, there's no more ATP being produced, right? So, and if there's no more ATP being produced, that means the muscles stay in a state of contraction because you have the cross bridges being formed. So you needed ATP to form the cross bridges, but now there's no more ATP to pull apart those cross bridges. So that means that the muscles are in a state of contraction. So this is state of rigidity. Eventually, all this comes apart, deteriorates, and then there's no more rigor mortis. Okay, so the troponin and tropomyosin, they contribute to the movement of the thin and thick filaments, right? Because of the calcium. So the calcium then on the tropomyosin, on the troponin, sends a signal to the tropomyosin that moves and exposes the actin binding sites on the thin filaments so that the myosin can bind. So here you have the explanation for rigor mortis. And it only stays like this for 3 to 4 hours after death, because then after 24 hours, everything deteriorates and the cross bridges come apart. No more cross bridges. Okay, so here are the summary of events that I explained. Acetylcholine binds to the receptor. Sodium enters and creates what we call depolarization. So when it goes from -90 to -60, or from -60 to 30, that is known as depolarization. Then you have the T-tubules with the dihydropyridine, which are receptors that are sensitive to the change in voltage, going from -90 to -60. So then they open the calcium channels on the sarcoplasmic reticulum. These channels are voltage-gated calcium channels. I will answer your question soon. And this increase the cytoplasmic sarcoplasmic calcium levels. The calcium binds to troponin, moves tropomyosin. This allows the sliding filaments. You need heat. And then you do the power stroke, the contraction. So the question is, what about what you were saying with Alzheimer's? Does that have anything to do with muscle memory? No. How would acetylcholinesterase affect muscle memory like how it affects the hippocampus? No, it has nothing to do with muscles. It's only neurons, because acetylcholine is a neurotransmitter also for neuron-neuron interaction. Here, you have a neurotransmitter where there is a neuron-muscle interaction. This is what I'm describing now. For the memory, you have a neuron-neuron interaction. And that's what allows for memory to form. It's this neuron-neuron interaction, where in some cases, the neurotransmitter is acetylcholine. So the Alzheimer's scenario has nothing to do with muscle. There's no muscle memory. It has to do with neuron-neuron interaction. Okay? I hope this is clear. Okay. Good question. Okay. So now you go from the action potential generated by the opening of sodium channels. Then you have calcium release from the sarcoplasmic reticulum. Then you have sliding filaments, and you have the development of tension. So here you see the muscle, the biceps and the triceps, the humerus, the radius. So they're attached to the bone. And now, because the muscle is, the sarcomere is decreasing in size, the whole muscle, because you have thousands and thousands of muscle fibers and you have thousands and thousands of sarcomeres within each muscle fiber, you have the contraction, and this develops tension. Okay, this is for you to see at home. Okay, so let's see some properties of muscle tension. The force that develops during this muscle contraction. How do the cells know which direction you want the muscle to move? Well, this depends on where the muscles are attached, right? Which bones. So you have muscle attached to the origin. That's the side that doesn't move. And here you have the insertion, which is movable. So depends on which bones the muscles are moving, right? Does this answer your question? Because you have some muscles that contract, other muscles that extend. I think I will answer your question better in another slide. Okay, so this tension that is developed by the muscle upon muscle contraction has some properties. Here I have three properties. One is that this is a muscle fiber with all the sarcomeres. So inside a muscle fiber, one cell, the response is a all or none. That means all the sarcomeres will shorten simultaneously at the same time, or none do. So this is in a single muscle fiber. It's an all or none. So all the sarcomeres within one muscle cell, one muscle fiber, will contract at the same time. Okay, so you could say, well, if that's true, how can you have like a movement that is so graded, right? So this is a movement that is not all or none, right? If it would be all or none, it would be muscle contract, muscle relax. No, but you can grade, you can cause the movement of the muscle in a graded manner. So how is this possible? How can you have graded contractions? So what this happens is that in the muscle, so in a muscle, you have many muscle fibers. So suppose that in this muscle, in the triceps, I'm going to say something that's silly, but let's say that it has 10 muscle fibers. Now let's say that the signal that is now regulating only affects 2 muscle fibers, but the strongest signal will activate 10 muscle fibers. So that's how you get the graded contraction. Depends on the number of muscle fibers that are being activated. So depends on how many muscle fibers, these are cells, are stimulated. So remember that one muscle has many, many muscle fibers. So the type of movement depends on how many muscle fibers are going to be activated. So we say are recruited for movement. So in the muscle, if you have 10 muscle fibers and only 2 are recruited, the kind of movement is going to be much lesser or smoother than if you activate 10 muscle fibers. Okay, so the other aspect that I want to bring up is that the tension developed by a muscle fiber is known as a twitch. So a twitch is the effect of one muscle fiber contracting and relaxing. So the twitch can be represented in a graph. So here, what I want to show you is this is a muscle fiber, and you see here many, many, many sarcomeres, right? So within a muscle fiber, all the sarcomeres, so here, this is the Z line, the thick and thin filament. So all the sarcomeres, when there's a stimulus, all the sarcomeres within a muscle fiber will contract simultaneously. So how do you get graded contractions? Is how many muscle fibers are going to be stimulated. Because if you want, let's say, if I want to hold, let's say this posted, I'm only going to recruit X amount of muscle fibers. Now, if I want to move my cup filled with water, it's much heavier. So I'm going to recruit many, many more muscle fibers. So fewer here, more here. So the grading, the strength, the tension developed depends on how many muscle fibers are going to be recruited. Okay, so now let's look at the twitch. So this represents the contraction of one muscle fiber. Okay, so let's start here where the muscle fiber is relaxed, nothing is happening. And now you have a stimulus coming in. So you have the motor end plate sending a signal. What's the first thing that happens? You need to have calcium released from the sarcoplasmic reticulum. So there's this period of time. So here you have milliseconds, and here you have muscle tension. So there's a lag period before the between the stimulus application and when the muscle fiber starts contracting. So this lag in muscle contraction represents the time that is necessary for calcium to be released from the sarcoplasmic reticulum. So when the calcium reaches a certain concentration, now you have the contraction, the all or none contraction phase. So all the sarcomeres are going to be activated and it reaches the maximum contraction. Then you need relaxation. So the muscle fibers now are going to go back to relaxation. When the calcium is removed, when acetylcholine is metabolized by acetylcholinesterase, so you have the contraction stage, relaxation stage going back to no stimulation. So now here you see the sequence of events that go on from the motor neuron to the development of tension by a muscle fiber. So the first thing that has to happen is the motor neuron to be activated. So it goes from -70, here it goes to +55 in the neuron, and then it starts repolarizing. So this is the depolarization. This is the repolarization of the motor neuron. So it requires a change in voltage. So this activation of the motor neuron then is transmitted to the muscle fiber. We're still talking about voltage in the muscle fiber. So it goes from -90 to +30 millivolts. So here's contraction, here's relaxation. So the opening of potassium channels. Here is the opening of sodium channels for the contraction, and here you have the opening of potassium channels for the relaxation. So then the next event, so you have the neuron activation, then you have the muscle activation in terms of voltage, and now you have the development of tension. So this is the latent period, which is the time required for calcium to be removed from the sarcoplasmic reticulum and reach a certain level. Then you have muscle contraction and muscle relaxation. So here you have changes in voltage, and here you have a twitch being developed, a change in tension. So now you know everything about how a neuron, or when you are giving an order to a specific limb or a finger to move, it goes through these three stages: neuron activation, muscle activation, and then the development of tension. Okay, so you have different types of action potentials being activated. So now you know from excitation, the coupling, as we started the class, the coupling of excitation to contraction. So you have the first action potential, number one, that's being developed at the motor neuron. Then you have an end plate potential that is developed at the neuromuscular junction when the axon terminal sends an action potential to the muscle fiber. First at the motor end plate, then the propagation along the sarcolemma. That's the action potential along the sarcolemma. And then you have the development of tension, the twitch. So you have these events leading to the development of tension in the muscle fiber. Okay, now the length of the twitch in terms of time, milliseconds, varies from some muscle to another muscle. For example, an eye muscle responds very quick, so it contracts and it relaxes. So the twitch has a very short time. So if you think about the eyes, all these muscles in the eyes, they have to move in a very, very precise manner to be able to focus. It's not like a muscle in the arm. You can move it a little bit more, a little bit less, doesn't matter. But the eyes, the muscles that control the eyeball, the movement has to be very precise and very quick. So the fibers here, you're going to see that there are different types of muscle fibers. So you have the twitch is very short-lived. Now this is the twitch for the gastrocnemius. So this is a muscle in the leg. So you see that the twitch lasts longer. And then in the soleus, which is another muscle in the leg, the twitch goes on for 100 milliseconds. So the duration of a twitch depends on the type of muscle fibers. And you will see that why is it that one twitch is so short, another twitch is medium, and the other one is so long. So this depends mostly on the amount of oxygen that is available, the type of muscle fiber. Some muscle fibers, I will explain this later. So it depends on the type of muscle fiber. Is this similar to muscle cramps? No, muscle cramps is when you run out of ATP and you have a lot of damage. We will talk about this. Damage on the proteins, and you have a lot of lactic acid accumulating. We will talk about muscle cramp. This has nothing to do. This is a normal thing. This is not damage. This is just the extent, the time of contraction, of the twitch of muscle fibers that make up these specific muscles. Okay, so now there's a period of time where the muscles cannot contract. And this is known as the refractory period. What is the major factor that prevents the muscle fibers from responding to a stimulus? When the inactivation gate for the sodium channel is closed and no more sodium can go in. So remember, this is the representation of a sodium channel, and it has two gates, the activation gate and the inactivation gate. So when there's an action potential coming down along the sarcolemma, then you have this alpha helix of this channel that changes in shape and allows the activation gate to open. So the activation gate opens and sodium rushes in. So when the action potential reaches that plus 30 millivolts, now you have the inactivation gate closing the channel. So it doesn't matter. You can increase the voltage. You can increase the strength of the stimulus. You cannot have the muscle fiber respond anymore because all of the channels are closed. They're inactive because the inactivation gate is in position and prevents sodium from coming in. So this is very important because it controls the amount of muscle contraction. You want the muscle to contract so much that it can rupture. So the refractory period is very, very important. It allows the muscle to recover and to get ready for the next stimulus. So you see here, you're going to do, some of you might already have done, the electromyogram, which measures the change in potential on the muscle fiber. So you have electrodes placed on your skin that can detect the change in electrical current in your muscle. And what you're going to do is use different levels of strength, and you will see that the electrical current, if you just do it slightly or stronger or very, very strong, the change in electrical voltage is going to change depending on how much tension you're developing. So if there's a small current, it's detected by the electrodes and the signal is amplified and recorded. So you're going to do this in the lab. Okay, so what controls the strength of the tension that is developed by a muscle? So you have here three different factors. The frequency of stimulation, what does that mean? how many stimulus are applied per minute, let's say. Then the length of the myofilament. So remember, the myofilament contains the thick and thin filament. So how much the thick and thin filaments are overlapping controls muscle tension. And then the third one is the number of muscle fibers contracting. Okay, so let's focus on the frequency of stimulation. So you can have these patterns that are very beautiful and interesting. So if the stimulus, the application of the stimulus is represented by this triangle. So let's say that the stimulus is applied very far apart, 200 milliseconds. So that means that there's time for the muscle to contract and relax perfectly before the next stimulus comes. And all this depends on how much calcium is left. So for the muscle to relax completely, you have to get rid of all the calcium in the cytoplasm. All the calcium has to go back into the sarcoplasmic reticulum. So here you see a single twitch where the stimulus are placed in such a time frame that it allows for the muscle fiber to relax completely. Now this stage here is a trepa. We call it a trepa. This means it's that the stimuli are applied more frequently than here. So that means that there's going to be some calcium left over. So the muscle doesn't relax completely. There's some calcium left over. So when the next stimulus arrives, you get a much you get a stronger contraction because there's calcium available. And then if there's even more calcium, so you're going to increase the tension until it reaches the maximum. So this is trepa. The trepa is defined as the contraction of the muscle when the stimulus is applied at the time difference, at the time point where all the calcium is not removed, all the calcium that is not going into the sarcoplasmic reticulum, and there's a little bit left over. So when the next stimulus is applied, there's already some calcium there, so the contraction is going to be greater. So in wave summation, the stimulus is even applied quicker. So you see that there's not all the calcium is removed, so you still have the muscle fiber in certain level of contraction. When the next stimulus comes, it goes up and up and up until it reaches the maximum. So here, the difference between trepa and wave summation is the level at which the muscle fiber contracts. Relaxes. Here, you see that it relaxes more than here because here the stimuli are applied faster. And then you have a condition where we call tetans. So you're increasing, I hope you get the point here, that you're increasing the frequency of the application of the stimuli. So here it's going even faster than in wave summation. So that means that there's still calcium left over and the muscle fiber is not relaxed. And because there's so much calcium, then it reaches the maximum very quickly. That's sustained. Tetanus is a sustained contraction. And this is the maximum. You see the frequency of the stimulus is so fast that there's no contraction. And most of the muscle fibers contract by tetanus. So that means that the stimulus is applied and so frequently that the muscle fiber has no time to relax until it reaches a certain level, the maximum, and the maximum tension reaches the level when all the calcium that is in the sarcoplasmic reticulum is released. There's no more calcium, and therefore the muscle cannot contract anymore. the increase. There's no more increase. And here you see another version of this where you go from a single twitch to wave summation. And then incomplete tetanus, you see that the relaxation here is much is lower than here. So you increase until a certain level, and then complete tetanus, there's no relaxation because the stimuli are applied so close to each other. Okay, so that's the dependence on the tension on the frequency of stimulation. But it also depends on the length of the myofilament. What does that mean? That means that the length of the myofilament, you think about the sarcomere, how much overlap there is between the thick and thin filament. So if the overlap is very little, then the contraction is going to be very weak. The same if it's overstretched. So if you have overcontracted or overly stretched, the contraction is going to be much less than if you have an optimal overlap of thick and thin filament. So the length of the myofilament, which depends on the overlapping of thick and thin filament, also controls the tension. And finally, the muscle fiber recruitment. So I explained this to you, that the development, so this is one muscle with several muscle fibers. If there's only a few muscle fibers that are recruited, then the development of tension is going to be much lesser than if you have all muscle fibers being recruited. So the number of muscle fibers being recruited controls the development of tension. So you see here, this is shown in this graph. So here you have this muscle fiber that has this muscle that has 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 7 muscle fibers. So each circle represents one muscle cell, one muscle fiber. So if you're recruiting just one muscle fiber, the twitch is going to be very low. The tension developed is going to be very low. Now, as you increase the number of muscle fibers that are being activated, then the twitch, the tension developed is much greater until it reaches a maximum when all the muscle fibers are recruited by that motor neuron. So here means that the motor neuron recruited the 7 muscle fibers, activated the 7 muscle fibers. And this depends on the signal. So the same thing that I was giving you example, to raise this, this post-it, I'm recruiting much fewer muscle fibers than if I'm raising my cup. So you need much more strength. You need much more tension. You need many more muscle fibers being recruited to raise my cup than to raise the post-it because it's much lighter. Okay, so I hope this concept is, okay, so question is, so if the overlap is very little, or if the overlap is overly stretched, then the contractions are going to be weak. Yes. So there's an optimal level of overlap for the tension to develop. Right. The Henman size principle. Correct. Okay. So now we reach the stage of fatigue. So let's say you recruited all these fibers of the muscle. Can you keep recruiting all the fibers? No. Eventually, although you're recruiting all the muscle fibers, you will develop fatigue and the contraction will go down. That's the fatigue. So how do you develop fatigue? When there's a depletion of ATP. So there's no more ATP to form those cross bridges. The pH in the muscle fiber declines. Why? Because you have lactic acid. And you'll understand why do you have lactic acid being produced in the muscle fiber. And then the sense of pain. Remember, I told you, because all these proteins, myosin, actin, troponin, tropomyosin, then the titin, all these proteins that are components of the muscle fibers start to deteriorate. Okay, so you're also going to do this exercise, which is going to illustrate the fatigue. So you're going to hold on to a dynamometer. You're going to hold it very, very strong and see for how long. So you hold it in weak and stronger, stronger, stronger. So the strongest, where you recruited all the muscle fibers. How long can you keep the recruitment of all the muscle fibers? You'll see that although you're exerting a lot of strength, the muscle fibers are going to be tired. So the contraction dies down because of the development of fatigue. Okay, so the types of contraction. So there's two types of contraction, isotonic and isometric. And this is very easy to remember. Iso means same tension, isotonic. So if I'm moving this cup up and down, the force, the tension that I'm developing is the same, but the size of the muscle changes, right? And contracting, relaxing, contracting, extending. So isotonic contractions are those that the tension is the same, but the size of the muscle differs. So if it's concentric, it shortens. So if I go this way, so the muscle is contracting, this would be a concentric contraction. Now, if I'm in this position and now bring it down, the muscle is increasing in size, it's eccentric. So the tension is the same, it's the size of the muscle that is changing. It shortens, that means it's concentric. It's eccentric when it stretches. So let's say I'm moving this up, I'm contracting the muscle. Now I'm moving it down, I'm extending the muscle. So concentric and eccentric. Now let's say I have the cup or a wall and I'm pushing it and pushing it and pushing it in a way that the size of the muscle doesn't change, because this is not, this is immovable, and I'm pushing against it. The muscle size does not change, but the tension does. I'm increasing the force applied to the cup. So this is an isometric contraction. So here the size doesn't change, but the tension does. It's like you're pushing against a wall. The wall doesn't move, but you can push it stronger and stronger so that the tension is increasing, but the size is not changing. That's isometric. And isometric contractions are used in physical therapy. You can choose a certain muscle and you want to exercise the muscle, so you use isometric contractions. Okay, question here. Oh, calcium ion fatigue. So calcium is if you don't have enough calcium in your system. Now, what do you mean by force is stronger? Though I thought the amount of force always stayed the same. Well, that's you mean in isometric contractions, the movement, the size of the muscle is the same. Now think about you moving a muscle. You're not moving a muscle. I'm holding the cup in the same position, the same place, but I can just touch it lightly, or I can push it with all my strength. So the force, remember, the force changes depending on the amount of muscle fibers that are recruited. So the force can change. It's the all or none response is related to the sarcomeres inside the muscle fiber. But the number of muscle fibers can change. And the number of muscle fibers that are recruited will determine the amount of force that is developed by the muscle. Okay, so this shows the same principle. Okay, so when each muscle fiber doesn't relax 100%, there's always there's always a few muscle fibers that are that are contracted, and that's known as tone. So, like I'm not moving my hand, so most of the muscle fibers are contracted, but there's a few of them. I mean, most of them are not contracting, but there's always a few that remain contracted. And this allows for the development of a muscle tone. Like in the neck. So I'm not moving the neck, but for my neck not to go back or forward, that means there's always a few muscle fibers in the neck muscles that remain contracted. And that's the tone. It's the fibers that remain contracted. So this muscle tone makes the muscles firm, right? They're not flaccid. They're not like all without any force. And the firmness with the tone depends on the sustained partial contraction of the muscle fibers. And this is essential for posture. So if we're standing up, you could say, oh, there's no muscles contracting in a person that's standing up and is still. No, that's not true. For us to be standing up, we need a few muscle fibers to be contracted. If they're not contracted, then developed this condition that's known as hypotonia, where the person is, there's no contraction, the body is flaccid. Or if it's overcontracted, then you develop spasticity. So these two conditions are depending on the amount of tone that is being developed. So hypotonia, no tone, hypertonia, over tone.
Yeah, one more day. So what's on? No?
Okay. So now, let's see. Oops. So now let me talk about muscle atrophy. So remember, I told you in one of the lectures that the number of muscles doesn't change. Like if you're doing a lot of exercises and your muscles increase in size, it's not because you have more muscle fibers. It's because each muscle fiber, each cell becomes larger. Why? Because the myofilaments, there's more myofilaments. The same, if you don't move, then the size of your muscles decreases. Not because you have fewer muscle fibers, but because the muscle fibers are smaller. So muscle atrophy depends on the number of muscle, on the size of the muscle fibers. So if you don't use them, they atrophy. If you use them a lot, if you do a lot of exercise, then the size of the muscle increases, not because you have more muscle fibers, but because you have the more myofilaments and more myofibers. So the change in size, you have more myofibrils. Myofibrils are those cylinders inside the muscle cells. Okay, I think I'll stop here today. You have any other questions? Okay. Okay, see you Thursday. We'll continue with muscles. Okay, bye-bye, everybody.